Hello everyone. We need to start our fourth set of genetics problems, the dihybrid cross. Now what we've been doing up to this point is called a monohybrid. Mono, of course, meaning one, and hybrid meaning a cross or a mating. So when we do a monohybrid cross, that is a cross or a mating based on one what? One trait, right? So what we're going to look at now is called a dihybrid cross. Of course, di meaning two, so what we're looking at now are two traits, and of course, hybrid still means a cross or a mating. So instead of just looking at one trait, we're going to look at two traits simultaneously. So how these problems are slightly different is up to this point, we've said, okay, you take a purple flowering Pisum sativum and you cross it with a white flowering Pisum sativum. And this is one trait, flower color, and what you end up with is a 2x2 Punnett square. Very straightforward. You guys are doing very well with this. What we're doing now on a dihybrid cross is we're saying, okay, let's say that you have a purple flowering tall Pisum sativum, and you're crossing that with, let's say, a white flowering, also tall Pisum sativum. You're now looking at two traits. You are looking at flower color, and you're looking at plant height. The rules are the same, phenotypic and genotypic ratios, all of the guidelines and how you dissect the problems are the same. What's the difference? The difference is that the Punnett square, instead of being 2x2, is 4x4, right? This is no different than when we were kids and they taught us long division and they said, all right, how many times will 3 go into 12, right? Versus how many times will 3 go into 12,427. The rules of division here and the rules of division are here. The only difference is this problem takes longer, but the guidelines and the rules are the same. It's exactly the same with monohybrid and dihybrid crosses. The monohybrid cross, pretty straightforward. Dihybrid cross is also straightforward. It just takes longer. That's the only difference. So don't be intimidated by the length of these problems. You can totally handle it. So be sure that this information gets into your Cornell notes. We're going to check your notebooks in about 2 weeks. You will turn in your notebooks just like you've been turning in your homework. Fantastic job on that. You're taking the pictures of your notebook, you're scanning them, converting it into a PDF and post it. Please keep that momentum going. It's a fabulous job. So since we're dealing with sexually reproducing organisms here, we need to drop, should be an A, we need to drop some vocabulary on you, of course. It's biology, of course there's vocabulary. So, a gamete, you go, well, what is a gamete? A gamete is a sex cell. It's sperm in males and ova in females. Okay, so these are gametes. These are sex cells. People commonly call these germ cells. No, germs don't make you sick. What makes you sick is a bacteria, virus, fungus. In biology, when we talk about germ cells, those are gametes, those are sex cells. So let's take, for instance, let's do a big B and a small b. Again, don't forget to drop this into your Cornell notes. And let's see that the big B allele is for big ears. And again, I'm just making this up. I'm just trying to pick some silly characteristic that's fun. And so then little b would be small ears. Cool. And then let's talk about hair. Hair is always fun to talk about. So sometimes people have different amounts of hair in between their knuckles. And so let's say that big H is the dominant condition, is actually thin knuckle hair. And then let's make the recessive really thick knuckle hair. And this is nothing to worry about, it's just hair, right? So we're playing some games here that we're talking about the size of our ears and the thickness of the hair in between our knuckles. Fine. Well, you know that we carry 2 alleles for every characteristic. So let's say that I have big ears. Okay, so I would carry a big B. Now, just to make me interesting, let's give me a small b. Let's make me a heterozygote. So I am heterozygous for ear size. I carry a big B and a small b. And let's say that I have, let's say, thin knuckle hair. So I have big H. And let's say I'm also heterozygous for this as well. So here's me as the heterozygote, big B, little b for ear size, and big H, little h for knuckle hair. Cool. So once I reach sexual maturity, that's the whole point of puberty, is it converts you from a child who can't sexually reproduce into an adult that can. So when you go to form sperm and ova, you have your chromosomes, like such. And we know that we carry our chromosomes in what we call homologous pairs. So here's my 2 number 1s that I received from my biological parents, and here's my 2 number 2s. So now these alleles are actually physical pieces of DNA. They're actually genes that sit on the DNA, and when these genes are read, transcription and translation, it leads to the formation of proteins that causes either my ears to be big or my knuckle hair to be thin. That's how this works. DNA to protein to a physical characteristic or your phenotype. So since I'm big B, little b, I carry a big B on this chromosome and I carry a little b on that one. You literally attach the allele to the chromosome. And again, I'm not saying that the size of our ears is carried on chromosome number 1. I'm simply trying to provide you with an example. And so then let's say that the density of our knuckle hair is carried on chromosome number 2. And again, I'm big H, little h. So here is my genotype. Here's what I carry inside of my genes. Well, I can't give both of these chromosomes to M, so there's a process called meiosis that we'll talk about in a couple of weeks, where these chromosomes get pulled to opposite sides of the cell, because I can only give Emily half of these chromosomes. So when these chromosomes get pulled in a process called meiosis, and I form my gametes, what I'm going to offer Emily is 1 of my number 1s and 1 of my number 2s. And then, of course, in the other gamete, since I'm a male, that's sperm. If you're a female, then that would be ova. And here's chromosomes number 1 and 2. So since these 2 chromosomes are on the left-hand side, when these 2 get pulled to the left, I would pull big B and big H to one side. So here's big B and big H. So if this gamete was used in fertilization, what Emily would receive is the allele for big ears but thin knuckle hair. It's just as possible that this gamete could be used in fertilization. These alleles are pulled to the side, and you get little b and little h. And so what Emily would receive here is small ears, or the allele for small ears, and thick knuckle hair. Fine. However, there's something called random assortment, and how these chromosomes line up, it's not always the dominant on the left and the recessive on the right. It's completely random. So while this is possible, it's also possible that the little b chromosome ends up here and the big B here. So all I did was I took the top 2 chromosomes and I switched them. You see how it's big B, little b here? And then over here, I switched them. So now the little b's on the left and the big B's on the right. And here's chromosome number 1 and chromosome number 2. Well, in the process of meiosis, these chromosomes get pulled to opposite sides of the cell. And look how this changes what goes into my gametes. If I have the big B and big H that get pulled to the left, I would have little b and big H, and this would be number 1 and number 2. And again, these 2 chromosomes here came from here. When these 2 get pulled over to the side, these 2 end up in this gamete. Whereas if these 2 chromosomes get pulled over, what I would offer to Emily would be big B and little h. Number 1 and number 2. So look at how the combinations are different. Emily might receive big B, big H, little b, little h. She might receive little b, big H, or she might receive big B, little h. It all depends on how these chromosomes line up side by side before they're separated. So we started with this in the parent. You say, well, how do you decide the different combinations? Well, in math, there's something called the distributive property, where if you have something like 2 times x minus 3, if you wanted to expand that, you then distribute the 2, like such. And you get, what, 2x minus 6. You agree? Okay? When you're trying to figure out what alleles are offered to the offspring in a dihybrid cross, you go back to what the parent offers, big B, big H, little h. And what you do is you distribute. You go like this and like this. So what could I offer Emily? Well, if I distribute my big B, I could offer her big B, big H. I could offer her big B, little h. And you see these two combinations here. Big B, big H here. Big B, little h here. Then you come back over here and you distribute the little b. Now this is sometimes where students get confused because how you write this is little b, big H. Students go, whoa, 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 I thought the dominant always comes first. The dominant does come first alphabetically, okay? So since B comes before H in the alphabet, the little b comes first. So that's little b, big H, and then little b, little h. And so by distributing the b's, you see that these 4 combinations are the 4 combinations that you see here. So this is what goes on the outside of your Punnett square. So please be sure that all of this information is in your Cornell notes. And then on the next video, we'll start genetics problem set number 4 on a dihybrid cross. Have fun.